The landing of a common housefly is a precise mechanical event, a touchpoint that initiates one of the most prolific disease delivery systems in nature. To understand this threat, we must locate the fly within the biological hierarchy: phylum Arthropoda, class Insecta, and specifically the order Diptera. This group includes a range of species categorized as medically important. While they all pose documented risks to global public health, their methods of interfacing with human biology are functionally distinct. Entomologists divide this order into two categories based strictly on feeding mechanics. The blood-sucking pathway includes species like the sandfly, the blackfly, and the tsetse fly. These insects operate as direct biological vectors. Their mouthparts are designed to pierce human skin and extract a blood meal, allowing for the immediate transfer of pathogens into the host's circulatory system. Conversely, the non-blood-sucking pathway contains the flesh fly, the blue bottle fly, and the common housefly, Musca domestica. Lacking the anatomy to bite, these species utilize a multi-stage life cycle to drive two massive public health threats: parasitic myiasis and mechanical pathogen transmission. The absence of a bite simply shifts the danger. It forces the fly to utilize a different set of biological tools to interface with human pathology. This diagram outlines the four-stage life cycle of Musca domestica. We can use this model to isolate exactly where specific medical risks emerge. The cycle begins when adults deposit eggs in moisture-rich environments, typically decomposing garbage or animal manure. These eggs are creamy white and resemble small grains of rice. Depending on environmental temperature and humidity, they hatch within 12 to 24 hours. The resulting larvae, known as maggots, represent the first stage of clinical concern. Diagnostic identification in the field relies on recognizing specific larval morphology. Note the segmented white body. The anterior end is pointed and conical for burrowing, while the posterior end is broad. The maggot is biologically driven to live and feed continuously on decaying organic material. When an adult fly deposits eggs on a human host, frequently near open wounds or natural body cavities, this feeding behavior results in myiasis. Myiasis is defined as the parasitic infestation of living host tissue by these developing larvae. At this stage, the fly is a localized parasite causing physical tissue destruction, a threat profile distinct from the mobile vector it will eventually become. As the organism matures past the larval stage, the nature of the risk shifts once again. After feeding, the maggot enters the pupal stage to undergo metamorphosis. This transitional casing is barrel-shaped and typically appears brown or black. Once the adult Musca domestica emerges, its range expands from the static waste site to any accessible environment within its flight radius. Public health officials identify the adult housefly in the field by looking for a few specific anatomical markers. The body is light or dark gray, and the thorax is marked by 4 diagnostic longitudinal lines. Under magnification, the characteristic wing venation provides a definitive fingerprint for species classification. Recognizing these markers is critical because it confirms the specific mechanical transmission risks present in a local environment. The adult housefly is mechanically limited in its feeding. It lacks biting mandibles and cannot pierce human skin. Its mouthparts are adapted strictly for sucking liquids or ingesting minute particles. This anatomy drives the fly toward moisture-rich, contaminated habitats, like excrement, garbage, and sputum. While navigating these surfaces, the fly's legs and proboscis naturally accumulate massive pathogen loads. The transmission loop completes when the fly moves from the waste site directly to human food sources. Because the fly cannot consume solid food, it must liquefy it before ingestion. The fly regurgitates and vomits its stomach contents onto the solid food, using enzymes to break it down into a liquid. Simultaneously, the fly frequently defecates while engaged in this feeding process. This combination of regurgitation and defecation deposits the pathogens gathered at the waste site directly into the human food supply. This entire behavioral sequence is known as mechanical transmission. The adult fly's danger is functional. It serves as a transport vehicle linking human waste directly to human consumption. This life cycle diagram tracks the development of Musca domestica, pinpointing specific human health risks. Parasitic myiasis is bound to the larval stage, resulting directly from the maggot consuming organic matter. Mechanical transmission is executed by the adult stage, a consequence of its high mobility. Mapping these threats onto the life cycle highlights two distinct points for vector control. Control efforts can either target the decomposing organic matter needed for larval growth, or physically block the mobile adults from accessing food sources and sterile clinical environments. The housefly demonstrates that an insect does not need a bite to be a dangerous medical vector. Its anatomical constraints and feeding behaviors provide a functional framework for the rapid transmission of disease across global populations.